In this video, we're going to talk about oogenesis. So that is developing the oocyte, which is going to be the female gamete. This is different. Remember, in the male reproductive system, the sperm is starting to be produced after puberty or at puberty. Oogenesis actually starts before a female is born, then changes at puberty, and then stops at some point. So again, that's very different from male reproduction, where sperm starts being produced at puberty and then never really ends. The nuclear events, so this isn't like bombs, this is talking about the division of the nucleus, is going to be similar. So we're talking about meiosis. So they're going to be the same, meiosis 1 that we discussed and meiosis 2 with the male reproductives is going to be the same, all the steps. When they happen and what happens is going to be different. So we'll see similarities and differences between male and female reproduction. So first is the first step, same that we saw with the male reproductive tract. Excuse me, that we start with mitosis. So mitosis of the oogonium. That again is going to be a stem cell. So remember, in male it's spermatogonium. In female it's oogonium. So both of these are going to undergo mitosis. The difference is this is puberty on. This is before a female is even born. So during development, the oogonium, the stem cell, is going to be dividing, and it's going to divide and divide and divide to eventually where it's going to be another oogonium and a primary oocyte. Still going to have diploid, meaning it's going to have 46 chromosomes because it hasn't gone through meiosis yet. So each oogonium is going to produce another one again, so then it can separate another primary oocyte and an oogonium and continue on that way. So that's going to be again before birth. So this is completed where mitosis and the entirety of the primary oocytes are produced before birth. Meiosis 1 even starts. So again, this is a major difference because it freezes. So between the 3rd and 7th month of fetal development, so in the uterus, a female, the primary oocytes start mitosis, but they stop in the first phase. So they just begin it. But they still stop at prophase 1, which is the first phase of meiosis, until puberty happens. So these guys are frozen in prophase 1 until puberty. Once puberty starts to occur, we get higher levels of follicular stimulating hormone, or FSH, to start the ovarian cycle. So this is what is happening each month in the ovaries. We're looking at both the ovarian cycle and the uterine cycle because they're kind of coinciding, but different things are happening in each. So each month, follicular stimulating hormone after puberty is going to be stimulated to complete meiosis 1. So not just one primary oocyte, some of them are going to be kind of large enough and the follicles are going to change that primary oocytes are going to finish meiosis 1. When the primary oocyte, so you have the primary oocyte here, this with meiosis 1, the division of the cytoplasm is unequal. So what happens is you get what we call a little tiny polar body because we have to divide the nucleus. So it's got a little bit of nucleus and a tiny bit of cytoplasm. And then we have a secondary oocyte that has nucleus with half the amount. So this is going to be haploid, or half the amount. So 23 chromosomes are here and 23 are here because we have to split the nucleus. So that is going to be the secondary oocyte because it gets majority of the cytoplasm. The polar body is just because we have to divide, but we divide unequally and we just get rid of this. So it goes away. And then this is what we're going to go on to next. So this is just showing you again, primary oocyte. Remember all the steps of meiosis. So pretty much it's frozen in kind of arrangement of those tetrads. So we've replicated our DNA and they are all arranged in group of four. And so then it's frozen. Then to finish meiosis, we have half the chromosomes in this nucleus and half of this in this nucleus, and barely any cytoplasm. Sometimes the first polar body can complete meiosis too. Sometimes it doesn't, but we don't care because we just get rid of it. So the secondary oocyte has most of the cytoplasm and then half the chromosomes. So the secondary oocyte is not a mature ovum, so it's not necessarily being ovulated yet. And a major difference as well is meiosis 2 is not completed unless fertilization occurs. So if the oocyte is ovulated, it may never finish meiosis. Only if it meets a sperm and a sperm penetrates is it stimulated to complete meiosis. So if each month fertilization is not occurring, these oocytes never completed meiosis. They do begin meiosis too, and it's another frozen thing. So in the primary oocyte, it's frozen in prophase 1. The secondary oocyte is frozen in metaphase 2. So it completes prophase, but stops at metaphase. The secondary oocyte is released or ovulated. And again, it's frozen. If fertilization ever occurs, it goes and leaves the body or is degenerated and nothing happens. If fertilization, where there is sperm that comes in and fertilizes that secondary oocyte, then we finish meiosis 2. And we actually produce a second polar body because, again, we have to divide the nucleus. So we complete with a second polar body, minimal cytoplasm, but separating the chromosomes. Get rid of that. And we have a mature ovum. So the only time we get a mature ovum is after fertilization. So secondary oocyte is what's ovulated. And only after fertilization do you have a mature ovum because it's finished meiosis too. So again, kind of looking overall. So all of this is happening before birth, where you have the primary oocyte. It's going to be frozen in prophase 1. And you have stem cells until we have all primary oocytes. Then at puberty each month, some of these primary oocytes are going to finish meiosis 1 and become secondary oocytes. Only one of these is going to be ovulated, which we'll talk about later. And we have a polar body that, again, we get rid of just because we have to divide the nucleus. This is half the amount of chromosomes, so we're at 23. The secondary oocyte is going to be frozen in metaphase 2, so the second part of meiosis. And only again, fertilization occurs. Fertilization, there we go. Got it. Then we have meiosis 2 finishing. We have our second polar body that we get rid of, and we have the mature ovum. So looking at the ovarian cycle, so we know kind of each step what's happening with meiosis. This is looking at changes in the follicles. So each one of those primary oocytes is actually going to be surrounded by a follicle. So that's where the oocyte can change and grow, and meiosis can occur. Is actually just a little encasing or follicle that's in the ovary. So yeah, this, and then we have the primary oocyte or secondary. We'll show. When we're not in puberty and we have primary oocytes, it's called a primordial follicle. And about 2 million of them through mitosis have been produced. By puberty, only about 400,000 primordial follicles are still around. Because a lot of times they just kind of break down, and it's called atresia. Each month after puberty, so this is after puberty, FSH is going to stimulate the development of multiple primordial follicles or primary follicles. So primordial follicles, again, are going to be inactive until they're stimulated and surround the primary oocyte. And it's usually just a layer of simple squamous cells that are follicles. And we have clusters called egg nests in the ovary that are going to be just clusters of the primary oocytes in the cortex near our tunica albuginea. Remember that kind of dense connective tissue. When we go from primordial follicles to primary, so primordial is just sitting around, now we're going to primary. Those follicular cells that are going to start getting larger, and they're going to start dividing and go from one layer. So primordial is going to be primary oocyte and one layer of cells. Primary, you're going to start stimulation where you're going to get multiple layers of cells. So you have kind of rings around your primary oocyte. We haven't changed from primary to secondary yet. So the follicular cells that are surrounding the oocyte are now called granulosa cells. So we've changed from follicular cells to granulosa cells. We also have what we call a zona pellucida. That's going to be the cells that are going to be around the primary oocyte. So it's going to be around there. We also have thecal endocrine cells. So you can say again in the name, if we have endocrine, it's going to produce hormones. So we also have thecal cells that are around the follicle that are being produced and made that are going to produce estrogen. And both of these, so the formation of the follicles are actually going to primary follicles, are going to increase estrogen levels because both the granulosa cells and those thecal endocrine cells are going to make estrogen. So you can see primary oocyte here. Granulosa cells are directly kind of surrounding the oocyte. And then we're getting thecal cells on the outside. So we're getting just multiple layers. Then we get to the point where some of those primary follicles are going to turn into secondary follicles. This is going to get even more layers. And those cells are going to start making fluid as well. So we still have granulosa cells surrounding the oocyte, primary oocyte still. We have thecal cells on the outer section, so you see you're getting a lot more layers. But then we have one kind of layer that's directly surrounding the primary oocyte that's called our zona pellucida. These follicle cells, so the follicular cells that are going to be kind of surrounding the oocyte, are going to start producing fluid that's going to make pockets and actually separate layers of the follicle. So we're actually going to separate layers of cells into different ones, and we'll see what happens next. Then we get to a tertiary follicle. So tertiary follicle is going to be the last one before ovulation. One of these secondary follicles, so only usually, not all the time, but one in normal instances, secondary follicle turns into a tertiary follicle. We also call it a Graafian follicle. You might hear it that way. So what happens is we have a follicle area that's filled with fluid, and it's called the antrum. And that's where the oocyte's going to project into. So we'll see, and I'll show you in a picture coming up. Granulosa cells are still going to be around the secondary oocyte because now changed from primary oocyte to secondary oocyte. Formation of tertiary follicle, because that's what's going to be ovulated. So granulosa cells are going to be surrounding the oocyte and what we call the corona radiata. So we're going to see kind of follicle cells, should actually be attached here, and then we're going to see a bunch of fluid, and you'll see picture coming up. So this is kind of what I was trying to draw. You have the secondary oocyte because we've gone to tertiary follicle. It's completed meiosis 1, frozen in metaphase 2. And we've separated cells. So again, we have layers of granulosa cells right here that are surrounding the secondary oocyte. And then we have a bunch of fluid because of those follicular cells producing fluid. We have kind of a fluid-filled structure around the secondary oocyte. The tertiary follicle eventually gets to the point where ovulation occurs of the secondary oocyte, and it takes corona radiata with it. So showing you all of this section is what's going to be ovulated. So it's the secondary oocyte and these granulosa cells that are surrounding it called the corona radiata. So all of this is actually ovulated, and it's going to say into the pelvic cavity, because if we looked at the beginning kind of our anatomy, we saw the infundibulum and there was little fingers on the edge of the uterine tube. Those are kind of draped across the ovary, but not attached directly. So there is space in between. So technically we have ovulation to the pelvic cavity, but then it travels into the uterine tube. Not all the time. Sometimes it can just go free into the pelvic cavity, but most of the time it knows where to go. So this all of this, the production, so primary follicles to primary to secondary to tertiary, is all the follicular phase, and it's dependent on FSH, and it's going to be high estrogen too. So we're going to see that later. Now, once ovulation occurs, 10 to 14 days into the cycle, luteal phase begins. So at the luteal phase, we're still looking at the ovary. The tertiary follicles still exist. We just got rid of the secondary oocytes. There's still cells that are around. So we only did the corona radiata around the secondary oocyte, but we still had all that fluid fill and the many layers of cells kind of on the outside here. So these granulosa cells that are still around are going to large and grow as well. And their major hormone is going to be progesterone. Still, they're going to produce estrogens as well. So they're going to produce hormones. Progesterone is going to be a major hormone that's going to stimulate the thickening and the readiness of the uterine lining with glands and things. And we'll look at the uterine cycle later. So progesterone is preparing for pregnancy. It's preparing for fertilization and then implantation into the uterine lining. So corpus luteum is going to be where that follicle changes from the tertiary follicle to what we call the corpus luteum. The major hormone that it's going to produce is progesterone. It does produce some estrogens, but the major one we're looking at this phase is going to be progesterone. If pregnancy does occur, so if fertilization and implantation do occur, so if fertilization and implantation occurs, corpus luteum stays until the placenta is made, and then the placenta will take over making these hormones, and among others. So the placenta is actually formed later, so we need time for it to be made. It's about 3 months the placenta is made, so during that time, the corpus luteum is helping. If no fertilization happens, then we have the formation of the corpus albicans. That is where all of this degenerates and regresses, and it's scar tissue. And it's actually scar tissue made by fibroblasts. So no fertilization, it degenerates the corpus luteum in about 12 days-ish, depends on the cycle, that varies a little bit. And that's the end of the ovarian cycle, because then we start again, of starting with our primordial follicles or primary follicles and going on that way. So again, we're looking primary oocyte surrounded by one layer of follicle cells is primordial. Then we go to our primary ovarian follicle. We start thickening. We get more granulosa cells. We get thecal cells. Secondary ovarian follicle, we're still just growing, getting more. We start having a zona pellucida, so surrounding the oocyte and producing fluid to kind of separate things. We have the tertiary follicle, which is going to be one of the secondary is going to go in. If not, the rest of them are just going to regress and degenerate. So we get rid of them. Form fluid. We have a corona radiata, which is granulosa cells, some granulosa cells surrounding the secondary oocyte. And then we have ovulation. Then after ovulation, we form the corpus luteum, which produces mainly progesterone, but some estrogen. So you can see here. So here we release the secondary oocyte with the corona radiata here. We have the corpus luteum, produces progesterone, mainly, and some estrogen. And then if no fertilization occurs, then we get the corpus albicans.